Okay, so just to let you know how this session is going to work, if you have any technical problems, if you can't hear us or anything's not coming through clearly, please let us know in the chat. We will see your messages coming through from on that, and if you can't hear us, anything's going on, please let us know there. If you have any questions throughout the session, please let us know in the Q&A box. We'll be able to gather them up and answer them, or they might be answered as the session goes along. So what I'm going to do now is, shortly I'm going to hand over to Brenda, but we've also got Mike Sulu and we've got Rana Khalife joining us from the laboratories, and they're going to show you around. So once again, thank you very much for your patience, a very warm welcome, and I'm going to hand over to Brenda.
Okay, thank you, Kim. Sorry for the late start, and thank you for those who had the patience to hang around. We just wanted to welcome you to this open day for, I guess, the admission cycle for 2022. How fantastic. So what we're hoping to do today is give you a bit of an overview about the activities of the department, of the teaching that we undertake here. And really, it's a forum for you guys to ask questions and to find out a little bit more so that you feel well prepared when you come to write your UCAS applications. So there's three of us today, triple whammy. So we have myself. I'm an associate professor in sustainable bioprocess design. I run a lab called the Biointegrated Design Lab, and I'm also the admissions tutor for the undergraduate program here at UCL. And today I have some good company. So I have Dr. Mike Sulu. Mike, would you like to introduce yourself?
Yes, everyone can hear me. I'm Dr. Mike Sulu. Also a lecturer in the department, obviously, and my specialism is in one area of the bioprocess, but I also run all of the kind of experiential engineering education within the department.
Okay, we have a third biochemical engineer today, which is Dr. Rana Khalife. Rana, do you want to say hello?
So hi everyone, my name is Rana. I'm a lecturer in teaching. I teach the regenerative medicine minor and some courses in the new MSc of commercialization of cell and gene therapy.
Okay, thank you. So yeah, I guess we're all here today to kind of maybe share with you some of the different aspects of biochemical engineering that we're involved with. And yeah, as I say, to answer your questions about sort of life in the department. So let's get started. So one of the most frequently asked questions, I suppose, in these admissions kind of presentations is, what is the difference between biochemical engineering and chemical engineering, and what's the difference between biochemical engineering and biochemistry? So let's deal with that first things first. So we share a lot of roots with chemical engineering. So many of the fundamentals that we learn here in the department, things like mass transfer, fluid flow, the observations of physical phenomena, we have in common with chemical engineers. However, the difference is the systems that we apply these to. So for instance, you might be looking at the kind of energy required to mix a fermentation vessel, but we would be concerned with how the cells within that vessel are experiencing it. Do they have enough oxygen? Do they have enough nutrients? Are the cells dividing and replicating and things like that? So, and really in common with kind of biochemistry and biotechnology, we do look into the more fundamental aspects of molecular biology and cell biology, but it's very much in an applied manner. So we translate many of the discoveries that are made within life sciences, but we're really looking at the kind of scale up and manufacture. So we're very much on the kind of practical side of things here. So just to maybe show you or maybe illustrate it as a continuum, we work in sort of synthetic biology. So biochemical engineers might be making the constructs, we might be engineering the cell lines with a particular goal in mind. And we will work in conjunction with molecular biologists, biochemists, and biophysicists to understand the kind of physical production of various different molecules using biological systems. And then a large domain that we work in is the bioprocess engineering side. So how do we scale this up into various different vessels? How do we purify our product? And how do we package it safely or distribute it to the end user? And then if it is a pharmaceutical product, then we would interface a lot with clinicians and other people in the health sciences to understand and evaluate the efficiency of the product that we've made. So we really combine all of these aspects of biology, chemistry, and engineering to produce useful products. And I think the most kind of a wonderful example of biochemical engineering in action at the moment is the rapid production and manufacture of vaccines. And we all know kind of how important that has been and how vital it has been that those can be rapidly manufactured in a safe manner. And that's really the role of biochemical engineers, is to work with the people who've discovered the vaccine and to work out how it can be made and then how that can be made safely in addition, so that patients can receive it in the correct form. So we are the first university to teach biochemical engineering as an undergraduate subject, and we are probably the only ones to teach it in this manner. We were a research department right up through the sort of 60s when we began an MSc program in this interdisciplinary field of biochemical engineering. But UCL has really had a vision from the get-go that biotechnology will be an enormous part of our futures, and from this reason, they built in the 1990s the Advanced Center for Biochemical Engineering, which is one of the largest types of facilities of its kind as a teaching facility within a university. And our undergraduate program is just over 20 years old. And in fact, Mike, Sulu and I are sort of one of the first cohorts of graduates from that program. And we studied here back in 2000. I think because we are unique, it often students ask the question of how to manage this on a UCAS form. So if you're applying for biochemical engineering, quite often you might be applying either for chemical engineering subjects or perhaps biosciences or biochemistry, or perhaps you might be thinking about it as an insurance choice if you're applying for medicine. All of those things are fine. So the way that we will look at your UCAS application is we will look at your personal statement. And what we want to understand is, do you have an interest in any of these areas? And if so, we will evaluate that on a case-by-case basis. So if you're interested in chemical engineering, please don't feel obliged. You want to have all of the offers possible on your UCAS form. We will still accept personal statements that are geared towards either chemical engineering or biochemistry, because we understand that we are a unique program, and that's the reason. So in terms of our teaching staff, we're a young department. You can see Gary Lai here is up in the top left-hand corner as our head of department. We're a diverse network of people who are all active in research in their own ways. So whether it's working on protein chemistry, microfluidics, synthetic biology, other aspects of process engineering, vaccine development, nanotechnology, we have a huge and very varied sort of spectrum of activities that we work on. But I suppose what we're all united in is this idea of translating discoveries and making realizable products. But we all have our own unique research agendas. And that's something that we try and feed into our teaching, because this is a field that's developing all the time. Every year new things are discovered and technology moves on. So it's really good that we are all research active and that we're able to kind of feed in and communicate the latest innovations from our space, even that are coming out of our own laboratories into the teaching program. So let's have a little bit of a think about what you might do while you're here and what you might learn. So in terms of studying biochemical engineering, and the same goes, I use biochemical engineering as a term, this is the department, but I do mean for bioprocessing of new medicines and both of the streams on that program too. What are we actually trying to achieve here? So we want to answer some key questions if we're thinking about products of biotechnology, whatever they might be, is how will I make it? What do I need to grow? How much can I make? How much do I need to make? Is it a rare kind of drug or is it a biofuel? The volumes will be dependent on the end use. How can we ensure that the product is safe, that it's reproducible every single time? And the key question is, how much will it cost? And that's, that will dictate how widely it can be adopted. So we are typically leaders in our field. We are responsible for dealing with the budgets. We're responsible for managing the risks and making sure that we have contingency plans in place. And that's really what we're looking for from you, is actually who will be the next generation of leaders in our field. So let's take you through some of the terminology. I see Mike's got his lab coat on, so he's down in the pilot plant and he'll be able to show you this in just a second. But let's talk some terminology first of all. So many of you will maybe have learned about recombinant protein production in biology. And if you haven't got biology, please don't worry. You don't need to leave the webinar. It's not a prerequisite actually for the engineering stream. But let's talk about this a little bit more. So imagine we were making a vaccine product and we had engineered a yeast cell to be able to produce this vaccine. And this is actually a real life example from a kind of universal flu vaccine project that's ongoing in the department. So you have genetically engineered your cell to produce this protein, and you have grown it in the lab in a very small scale, maybe in a few milliliters in a shake flask. That's obviously not going to be enough for all the patients that you need to administer to. So you need to start to scale up. And as you scale up, you will encounter different physical challenges. So how do we mix a vessel so that the cells maintain the correct level of oxygen? How do we supply them with enough nutrients? If they grow and metabolize, will they change the pH? How do we keep that regulated? And as we scale up, these things become more and more complex. And this is what we call upstream processing. So this is all about the cultivation and growth of cells. Once we have our kind of vat of maybe sort of 10,000 liters of our yeast is in a broth. So we're going to need to separate the cells from the media that it's growing in. So we might use something like a centrifuge, and we need to understand how fast to operate that centrifuge so we don't break the cells open and lose our product. If our product is intracellular, so it's inside the yeast cell, we're going to need to break the cell open, and then the fun begins. So obviously within a cell, you have DNA, you have other proteins, you have other organelles even, and your job is to distinguish what are the chemical properties of my product that make it distinct and different from the other aspects of the sort of cell contents and the cell soup. And then you will use that knowledge to be able to separate it out, either on the basis of size, of electrostatic charge, maybe of other kind of physical properties that it has. And once you have been able to separate it, then you can package it and then sort of distribute it to the end user. So you can see here that you need to understand a bit of biology to grow the cells. You need to understand chemistry to separate the product. And you also need to understand the engineering aspects to look at the physical apparatus and how that works. So you can see it's very, very interdisciplinary, and it's really well suited to people who enjoy being able to apply the various different subjects to a kind of problem solving exercise. So what kind of things do we teach, and what kind of things do we apply this upstream and downstream business to? So many of our staff work on the production of biopharmaceuticals. So this could be vaccines, but it could also be antimicrobials, antibiotics, but also immunotherapies. And in the case of Rana, cell and gene therapy even. And she'll tell you more about that in a second. We might be looking at the cultivation of human cells into tissues in the example of regenerative medicine. Or you might be looking at something which is more related to sustainability, which is, I guess, a little bit more in my remit, which is looking at kind of bio-based chemicals, biofuels, and biomaterials, and how we produce those. And in the future, it could be anything with synthetic biology. It might be bioremediation of pollution. It could be the production of novel materials, pigments, dyes, foods, even. All of these things depend on an understanding of scale up of biological systems in this really applied manner. So just some examples of the work that we're doing. We have the UCL Plastic Waste Hub, which is looking at how we will maybe be able to manufacture enzymes and apply this to plastic waste to degrade it and actually upcycle it into useful monomers, which can be then turned into other products. Looking at how we might manufacture personalized medicines, which is part of a future manufacturing research hub. In my case, we're looking at how we can apply algae to clean wastewater from heavy metal pollution. And finally, we have a huge effort with Oxford University on how to manufacture sufficient vaccines, which I think will be probably a defining characteristic of this next era of biochemical engineering and how we produce and can adapt our manufacturing processes rapidly when new vaccines come online. And you're very welcome to have a look at any of those activities if you're interested in, the kind of future of where our field is going. So what is our ethos here within the department? We want to really bring to life the education experience. And I will be quite frank here as admissions tutor. Much of what you can learn at university, you can learn online. As you know now, online learning is amazing. There's a diversity of information. So what do you come to university for? What is the value of a university education? In our case, we believe it's very much aligned to kind of small group activities, practical activities, and being able to build a network around you, and also to be able to participate actively in research and get involved in things. That's actually the meaningful side of education. And I think that's where education is going in the future. I think Mike will be able to talk a little bit more about the experiential side of things. But UCL has something called the connected curriculum, which emphasizes that even at undergraduate level, you should be empowered to take part in research and solve problems that are, actually real life ones as opposed to constructed ones. That's what gives you confidence, and that's what enables you to apply what you have learned in the theory. So we have a number of mechanisms to do that through what we call scenario weeks, which are 1 week long exercises where you will apply the theory that you've learned in the modules to a certain defined problem. And other kind of practical and workshop based activities that you will do with other engineers within UCL. So let's get to some of the details about the programs themselves. So we've covered the kind of ethos of how we teach, and now it's maybe a useful point to kind of look at the 3 different pathways that you have within our department. So if you're here and you are studying maths at A level, you are eligible for the kind of BEng and MEng route. We'll cover the subjects in a moment. And for those of you who don't have maths, but you have maybe a mixture of the other sciences, there are the other 2 routes in the BSc bioprocessing programs. Now, they all have different emphasis, but the whole point is that what we really believe in the department is that biochemical engineering is not just solely an engineering degree. There are routes for other people who may be more interested in the biological sciences or the business and management side of things. So that's why we have these various different pathways, but they share many modules in common. It's just a little bit in the kind of emphasis of where each program lies. But we have, as I said, we pioneered the first biochemical engineering programs, and really our graduates are known worldwide, and there's a huge alumni network which you can tap into when you graduate and when you leave. So before you go anywhere, let's see what you're going to study when you come here. So on the BEng and MEng routes, so these are co-taught for the first 3 years, and after year 3 you will choose your pathway if you decide to continue on to the final year of the MEng. So it's what's called an integrated master's program. So the first year is really an introduction to the core subjects, so things like thermodynamics, maths, biochemistry. Everyone will be going at different speeds, so people with biology background might find biochemistry easier, people who've come with physics might find thermodynamics easier. The point is, at the end of year 1, you all arrive at the same place, which is ready to kind of look at the application of these subjects. So in year 2, you start to take on more advanced courses and discipline-specific materials. But what you will also begin to do is take what's called a minor subject, which is a block of 3 modules that will run in parallel to your main biochemical engineering degree. And that will give you a sort of specialism. And you can choose things like programming. You can look at more advanced chemistry. You can take a language. You can look at environmental engineering. There's a whole list of subjects that you can choose, and you will begin to build a kind of companion to your main program in biochemical engineering. And that can also give you a very interesting edge. So in the 3rd year, what you will do is you will take a design project, which will invite you to design from scratch the manufacturing process for perhaps a target pharmaceutical or a drug or a therapeutic. And you'll also then finally take even more advanced modules in sort of computer-aided design and more molecular biology. And in the 4th year, you have your integrated master's year, if you choose to stay on the MEng route, and I will go through those in a second. So you can have a little look on the UCL Integrated Engineering Program page for a bit more information on exactly how we interact with the various different branches of engineering throughout the years. So for the MEng route in year 4, you have a number of different options. So first of all, you're able to take the final year with chemical engineering if you want to have a bit of an insight into maybe sort of more hardcore process industries as well, and look at more kind of advanced chemical engineering principles. 2, you can take the final year in biochemical engineering itself. And what that will mean is you will undertake a research project, an extended research project within one of the groups here at UCL. So perhaps you're very interested in cell and gene therapy. You can spend a number of months working with a lab group here with PhD students here on your own individual research project within the department. Thirdly, we have an MEng program with study abroad. So that means that you can go to any of the partners that UCL has that has a kind of comparable kind of process engineering department. So we've had people go to Canada, to McGill, to Melbourne, to Singapore. I was fortunate enough when I did my undergraduate, I went to Caltech for my final year. And it's a chance to kind of travel and also maybe gain a bit of a broader perspective into other approaches to biochemical engineering. Fourthly, we have a route of MEng in business and management. So again, that will really focus on the kind of commercialization and business planning around the biotechnology sector. And finally, which is a really, I just think, an amazing program, which has just taken off incredibly, which is our MEng and year in industry. So most engineering departments will do a kind of sandwich year in industry. The feedback from our sector was that these students weren't quite ready. And if they were able to do it after their 3rd year, they would have a much better level of understanding and would be able to undertake maybe more interesting research projects or placements at industry. So we took this on board and we developed a final year program where students will spend the majority of the time at their industrial partner. They will be paid to do so. And they will come back to UCL for a number of short courses during that year. So during the year, they'll be taking a research project within the company. So we've had people go to GSK, we've had people go to Eli Lilly in Ireland, to Lonza, to a number of sort of the big kind of biotech and bioprocessing companies within the UK and Ireland. And we're sort of expanding that network all the time. So it's a very competitive program because you have to obviously interview and be selected. But the experience of students has been fantastic, and many people leave with an offer either of a job or of funding for a subsequent PhD, and the response from industry has been absolutely terrific. So I think this is an innovation that's really, really worked for us. In terms of the BSc, so they are quite similar in terms of the pathways, but they're slightly different in terms of the emphasis. So with the BSc Bioprocessing, the science and engineering, this is maybe more geared to those of you who've got biology and chemistry at A level or IB equivalent, who want to maybe go a bit more deeper into sort of biochemistry, molecular biology, genetics. But this is quite a flexible program in that you have a number of credits of optional modules that you can take from the kind of wider course selection at UCL. You will have this core of biochemical engineering through the modules that you will take in our department, but you can then supplement that with maybe more biological sciences as well. In the 3rd year, you will also do a design project, albeit slightly shorter than the engineering route, because in the second part of your 3rd year, you will take a research project within the lab with a supervisor here. So you will, for instance, be working with a lab group here on a kind of research project, which you will get to choose. The same goes for the BSc Bioprocessing with the business and management, but many of the optional modules are supplemented with courses from management, accountancy, and the School of Management Sciences. Again, you will build up your core of biochemical engineering, but you will be supplementing that with things like law and sort of business planning and other modules which are much more related to the kind of business and management side of this industry. And finally, you will also take the design project with a research project in your final year. So again, these degree routes are just as good. I mean, they are fantastic degree routes in and of themselves. They just offer a slightly different flexibility to the engineering route. So as I mentioned, we really enjoy learning by doing, and we're so fortunate to have these fantastic facilities here at the Bernard Katz building, where we can really make good on our promise to kind of deliver this idea of the connected curriculum. And here you can see just some of the student activities that people get involved with. So we have the Igem team. So Igem is the International Genetically Engineered Machines competition. Every year we field a team. Every year we win a medal. It's actually very good. This is where students will work in the lab for a summer on a genetic engineering challenge. So in this case, they're looking at a breast cancer therapeutic, and then students get to go to Boston for the Jamboree at the end. We have some students presenting work that they did in the second year, scenario week on, in this case, they were looking at how to make biofuel from waste coffee, and they presented that in Parliament. And here you see Julia, who is one of our BSc students, and she was awarded one of the Royal Academy of Engineering scholarships for her work. She's been really interested in plastic degradation and spent some time in the summer doing internships here with John Ward. So if you would like to join our degree, the grade requirements are pretty straightforward. We do actually have some lower offers for students who, contextual offers for students who might be eligible. That's something you can read a little bit more on the UCL website, or you can contact me about. But in general, the grades required are 3 A's. For the BEng and MEng, it is imperative that you have an A in A-level mathematics, because mathematics is co-taught and through the IEP program. There are flexibilities around the other subjects that you have. So for the BEng and MEng, you can have any 2 from the 3 sciences. From the BSc with Science and Engineering stream, you must have biology and chemistry. And for the business and management, it's actually completely flexible. You must have at least one of the sciences, but you may be taking economics or some other subjects as well. Again, a lot of this is, if you're an overseas student, the equivalents will all be on the UCL admissions website, but please get in touch if you have any doubts about what subjects or grade requirements that you have. So thinking about life beyond the program, obviously when you're at university, you want to enjoy your time to the full, but you also want to maybe put in place a network to help you as you move from academia into industry. So what we do on our part is we use our network of contacts, our alumni network as well, and we invite them for things like this poster presentation here that you see with the kind of third-year students on their design project. And we have a huge kind of spectrum of companies and people that we are in contact with through research projects, through maybe former students, alumni, and other industrial partnerships, where we invite them to the department. So they come and see your work, interact with you, and you get feedback from people who are really working in industry, because it's so important in our field that you have a realistic appreciation of what is actually done in real life whenever it gets to these companies. And just to really say, and to leave on this note, is that this is a growth sector. I mean, we hear all the time about companies that just can't get enough biochemical engineers. I think certainly the recent kind of vaccine production, the pressure to be able to deliver this has really meant that, people are recruiting biochemical engineers like crazy. Rana can maybe comment a bit more on the cell and gene therapy sector specifically, but it's one of the biggest skill shortages that we have. So our employability, is enormous. You learn a lot of transferable skills on an engineering program, which will obviously take you to a number of sectors, whether it's finance, whether it is enterprise, innovation, whether you stay in the sector itself or whether you go on to further study in academia. It's a growth field, and really, it's an exciting time to be a biochemical engineer as this industry is taking off, but also diversifying into so many new areas like functional foods, cultured meat, biomaterials, and all kinds of things. So just to close, why do I think you should study a biochemical engineering degree at UCL? I think it is a really exciting degree. It's a field that is, I don't want to say it's emerging because I think we're already established, but it's a field that's growing. If you're interested in becoming a chartered engineer, both our BEng and MEng programs are accredited by the IChemE, so you can become a chartered engineer following graduation. You will join a global network of people really making a difference in the biotechnology sectors, who are really contributing to access to affordable healthcare, who are really making kind of pioneering breakthroughs in biotechnology. I think this is a great time to be part of this field. But finally, and most of all, thinking of the very short term, it's to spend your time in a small department that's very friendly, that gives you lots of opportunity to engage with cutting edge research, to get hands-on experience, and just to go beyond the idea of learning via lectures, that you actually get to learn by doing. And I think, as I say, that's the key to being confident when you graduate and to being able to sort of navigate the world afterwards. So I'm going to stop here. I think that is my final slide. Oh, apart from my email address. So yeah, you can always reach out to me as admissions tutor if you have any questions. We've put lots of stuff on our website, but I know there's always one or two people who've got unique things every year. So you can always drop me a line. And yeah, you can read a little bit more about our activities on the VAX Hub and the Plastic Waste Hub if you're interested in those subjects. So I'm going to stop sharing because I think, by the miracle of technology, Rana and Mike are both in laboratories right now. So yeah, Mike, do you want to kick off? Where are you right now, and what are you doing?
That's a good question. I am in the pilot plant, and
what is so yeah, maybe you want to explain what exactly is the pilot plant for those that haven't heard of a pilot plant before.
It's so I guess within academia, it's a relatively unique facility for us to have one, but within industry, they're kind of commonplace. I think we might be the only university that's got a complete pilot plant, but essentially what they are is a scale of operation which you would use to test out industrial processes. So if you were industrial manufacturer, you would have a lot of things that you would work at on a bench scale, so tiny volumes, and we're looking at, you know, around liters. And then you'd have an industrial scale, which would probably be hundreds or thousands of liters. And in between those, if you wanted to try and validate your process to make sure that your process would continue running as it would industrially, you have that intermediate scale called the pilot scale. We have an entire pilot plant here at UCL, and it usually kind of, we're looking at working around tens of liters of volume. So we have fermenters that range from under 1 up to 100 liters, and then we have the rest of the process that can kind of run with that kind of volume of processing. If we think about what Brenda's already told us, we are big proponents of learning by doing, which is basically what experiential engineering is. And we are, because for more than one reason, we wanted to be able to use our kind of unique facilities to help our students learn and improve their learning. And if you look at the educational literature, which we did, it basically says students will learn in more than one way. And some students are fine just learning theory, and they can apply that theory to practical situations immediately. But some students also need to have the practical element, and some students will only really learn through the practical element. And it helps kind of synthesize and synergize like knowledge, so that it basically just improves the way that you learn and enhances the way that you learn. So we do that in a variety of ways here at UCL. There's, I guess you could broadly split the kind of practical education into different groups. So we generally start off by teaching you how to do things. So if I flip my camera around, can I flip my camera around? Yes, I can. I'm sitting in front of a fermenter. And this is a tiny kind of 250 ml bioreactor. And this is where, as Brenda showed you the entire bioprocess, this is where you'd be growing the yeast cells, for instance, to make like a universal flu vaccine, for instance. We would teach you how to use this. And then that would be one kind of practical learning. Another form of practical learning around not just learning the basics of using pieces of equipment would be trying to work out what you would do as a practicing engineer. So we think about the scenarios you would be in. And as a practicing engineer, you might be asked to design some experimentation that says, if you run a small scale fermenter, what would that look like in an industrial process? And you have to then use some of the information that you've learned through theoretically, and also use some of the information you get by doing some experiments to try and theoretically work out or empirically work out what that large scale process would look like. And the other form of practical work that we do is all about the things that Brenda's been talking about, which is research. So you then get more kind of free reign. You're looking at potentially an entire process that interests you, or just one part of the process that interests you, and looking at trying to work out new forms of information or new knowledge based on the experiments that you run. So a good kind of experiment you might want to do is you take that small fermenter, and instead of running with that, you would be thinking about, what would I need to do to make it run the same as one of these? And so you'd run the small fermenter again and again and again. Then you also then think about theoretically what that would look like large, and then you run it in the large scale and think about whether or not the two match. And if they don't match, why don't they match? So as you will find out if you attend, one of the things that engineers will be doing is scaling up. My favorite part of the process is the fermentation. And for me, that's because I think it has the most different elements of science within it. So you need to know some biology because you'll be growing live cells. You need to know some physics because it's all, and the engineering physics is all about mixing two-phase systems or transferring mass or heat to different locations, or ensuring that mass and heat are transferred equally across a fermenter. But that isn't the entirety of the bioprocess. So I'm going to keep walking, and little Crystal made us dash to the pilot plant, to the rest of our pilot plant. And as Brenda showed you, which I'm going to show you more physically, after you've grown some cells, you need to think about what and where your product is. So if your product's in the cell, you're going to break it open. You break it open using a homogenizer. Homogenizers, they're kind of adapted from the dairy industry, which they use them to homogenize fats in the milk, so the milk is one smooth liquid and can be frozen. It can look like this. It basically works by you getting a sudden change in flow rate and pressure, and that change in flow rate and pressure within the unit operation is enough to disrupt the cell wall and leak the contents of the cell out. So if your product, which might be a therapeutic vaccine, is inside the cell, you're going to use one of these to break it open. You can also break it open chemically, but if you're going to break it open mechanically, you'd be using one of these. Interestingly, this is also a homogenizer, so you can see they actually look very different, but they work in the same way. After that, you're going to want to think about the fact that you're now got a mixture of liquids, partly solid, and then you're going to have a product which will hopefully be soluble. If you're going to use, if you want to separate your solid from your liquid, you might use a centrifuge, which is one of these. A lot of people will have seen a benchtop centrifuge, which spins little tubes down, but which is great. But at the pilot scale, when you're using tens or hundreds of liters of liquid, you're going to use one of these because it'll be able to do it continuously. It works in the same way. Inside this bowl is a series of discs that spin super quick, and they separate based on density. So that's how you separate your solids from your liquids. You might also use a filter, and we've all seen a filter. Because I guess we've all seen things like a coffee filter. We would call those a dead end or depth filter. We also use a technique called tangential or crossbow filtration. And in that situation, what we have here is this white part would be the filter. And what you would get is a continual flow across the filter of the mixture of your solids and liquids, and then a sucking or a change in pressure across the filter, which suck the liquid away. The reason we do this is because you would have seen with a coffee filter, is eventually the coffee grounds build up on that filter, and that affects the efficiency of filtration. If you can get continual flow across the filter, you don't get any buildup of that solid material, which means your filtration stays continually efficient and you can filter better. And the last part is chromatography. So chromatography, I think those of you should know, because you would have done things like thin layer chromatography probably in a practical at school quite a while ago. Basically works based on affinity. So there you have a different affinity to solvents running through a paper, But what we have here is a liquid. I'm going to get close to it. You probably won't be able to really see it. Inside that white milky liquid is like lots and lots of little plastic or plasticky beads. The beads have been functionalized, and that means they've had their surface chemistry changed. So we can make each little bead either positively charged or negatively charged, and that will have the effect of either attracting negative or positive molecules to it. You flow the liquid through the chromatography column, and essentially, based on what the charge is or the affinity that or the chemistry you change on the bead, it will slow some things or capture some things at different speeds to other things. So sometimes it will be, say for the interesting one is for antibodies. If you cover the surface of these beads with antigens, the antibody will naturally stick to the antigen, and then you'll get everything else through apart from the antibody. Then you can release the antibody by changing the chemistry, the environment within the chromatography column. And that's how, kind of the last step in the bioprocess. I'm going to take you on through now our pilot plant and show you a few other interesting things that we have. And you get the joy of not looking at my face. We have a set of robot platforms, which are super interesting. They're all very similar, and they look like this. They're interesting because we realized a while ago that one of the areas in which errors can occur is the human intervention. So if you're going to be moving tiny volumes of liquid around a process again and again and again, it's very difficult for a person to do it repeatedly. So what we do is we set these up to do that kind of manual labor for us. And then we just look at the results that come from it. And the interesting thing about these is that these can effectively mimic anything that we've seen so far. So for instance, this is a 96 well plate, so named because it's got 96 wells in it. It takes like maybe 0.5 milliliter of liquid in each one. You could use each one of these as a different fermentation vessel. Then you're running 96 fermentation experiments at once. But what will then happen, of course, is when I showed you the small fermenter you put in your hand versus the 100-liter one, which was entirely made of stainless steel, is you're going to have different things to think about when you're thinking about what will each of these look like theoretically and physically compared to that big 100-liter fermenter. And that kind of leads to some of the kind of more complex research questions that you'd be asking yourself if you were doing the 4th year M-Engine biochem or the end of the 3rd year in the BScs, because they're the kind of things that you would be able to think up questions or have questions that you choose, research questions, and then investigate for yourself. So that's the end of the kind of mini pilot plant tour. I'm going to turn back to see me again.
Awesome.
And hopefully Rana will do the same upstairs.
Yeah.
So quick word to say that we're going a bit over time, but this is being recorded and everyone will be sent a link afterwards. So if you can't stay whilst we make up time, there will be a link sent afterwards so you can watch it at your leisure. Okay?
Yeah, but you're all staying. It's great. We love having you here. So we are going to go from level 1 to level 5 and join Rana upstairs in our funny little live broadcast. Rana, you're in the lab. Do you want to tell everyone a bit more about cell and gene therapy? Because we didn't talk too much about it today.
Yeah, sure. So hi everyone. Let me just briefly introduce myself and what I did as undergrad and PhD, and try to convince you why you should do biochemical engineering. So as my background, I did my BS in biochemistry. Maybe back home we did not have the concept of biochemical engineering. And then I did my master's in cancer research, and it was great. You publish paper. But then I came to a point in which, okay, you're publishing a paper, you're doing research, but how can you implement this in real life and like translate this kind of knowledge, this discovery to real life and help people? So I found out the biochemical engineering at UCL, the department, and I was happy to do my PhD over here. And then
Okay,
I've been teaching the regenerative medicine minor. So as Brenda mentioned, within the Faculty of Engineering, we have different minors. Our department offers 2 minors, one related to the math and then the regenerative medicine. So I teach more about the cell and gene therapy, bioprocessing, tissue engineering, stem cells, and the new concept of CAR-T. So I will explain a little bit. So I am a bit biased. So Mike said that fermentation is the best for me. It's like stem cells, growing stem cells are the best part of it. I hope Mike that won't hear that anyway. So stem cells for you, I'm pretty sure that you covered this maybe at school. There are cells capable of generating any type of your cells, like the embryonic stem cells. Now, there has been a lot of ethical problems after 1998 when they isolated these kind of embryonic stem cells. So the research has stopped until 2007, where the Nobel Prize winner, Professor Yamanaka, discovered how he can reprogram any cell type in your body to become stem cells. Now, there are a lot of clinical trials happening on stem cells, but the main problem, as Brenda mentioned, is the cost of the bioprocessing and trying to optimize these kind of bioprocessing. This is what I did during my PhD, and some of the PhD students are working on that. So how can we optimize the process of, let's say, take a skin cell from a patient, reprogram it to become stem cells, and then differentiate that into, let's say, retinal cells, and then give it to the patient. So we know each step. How can we optimize each step and then lower the cost of it? This is how we. This is why we do our research over here. Although we are a small department, we have a variety of research, as you can see. Even within the Regen Med or cell and gene therapy research, we have a lot of variety. So I do more of stem cells, of induced pluripotent stem cells. We have other people working on small particles like exosome. We have people working on different type of IPS. And the most important one is now the trend, which is the CAR therapy. So I don't know if you've heard of it. I just want to explain briefly about it and then show you some cells. So nowadays, with the new therapy for treatment for cancer, especially like leukemia or blood cancer, what they're doing is they take the blood of the patient, they isolate the T cells, which are responsible to fight cancer. They genetically modify it and then give it back to the patient. The process looks so easy, straightforward. The most important challenge in that is the cost of it. So do you know roughly, you can type on the chat, do you know roughly how much does it cost to give the patient 2 doses of this and then they will be cancer-free? Any thoughts on the chat? So the first 2 products that they went on market, it cost around 475,000. Esther, 1 million, you're almost there, $475,000. So the NHS refused at the beginning to use it over here, but then they come up with an agreement with the company, and the first patient, NHS patient who have received this CAR-T, he took it, I think, 2 years ago at the cost of £200,000. So this is the role of biochemical engineer. As Brenda mentioned, we want you to be the leader in the bioprocessing, and this is how we teach our undergrad students and even the master students, because when you go, there is a lot of shortage in bioprocessing. And I went to Catapult 2 years ago, and they were saying that we have a shortage. At least we need 200 to 300 people with bioprocessing backgrounds each year to fit in this thing. Okay, so I'm gonna show you a little bit the lab now. Unlike Mike, we do over here in this level only cell culture, and we do it in a smaller scale, and I will show you the larger scale in a bit. Okay, just like any lab, you already know, like, this is a light microscope to observe the cells. We have more advanced one in which you don't use the eyepiece, you can use this kind of a nice technology. Now, what Mike showed, that you can use the bioreactor to grow the cells, for us, because we are afraid of any contamination, especially bacteria or yeast, we grow the cells, or we handle the cells in this kind of thing called a hood, in which the airflow will go to make everything sterile inside. So this is how we put some nutrients to the cells. And we have these kind of things. Does anyone have any idea what are these? What are these called? Okay. So these keeps the temperature 37, 5% CO2 and 20% oxygen, just like in our body. And we call these stuff kind of incubators. And as you can see over here, we're dealing with a smaller scale of experiment. Once we optimize the bioprocessing of the small scale in each step, we go into a larger scale. And as you can see, like the red things are the nutrients for the cells, and you can grow the cells in a smaller scale, which is T75, which is 75 centimeters square, up to a smaller one with like 24 well plate. Once you optimize this kind of process, you can move to even bigger one, and you can use this kind of a shaker flask, in which it's like, I think it's 25 ml now here in the flask. So you're optimizing the scaling up to produce more of these cells in the lab. Now, this is one part of the lab. I'm gonna show you another part in which once you optimize the cells, you can even grow in a larger scale. So we have even a larger shaker flask where you can grow your cells. Of course, this needs to be put in the incubator at 37 degrees. Just bear with me 1 second. Now, once this optimized, you can even scale up furthermore into a 250 ml. So I can see the bioreactor over here, the capacity is 250 ml. And over here, we're trying to optimize the expansion of these T cells for therapy. As Brenda mentioned, we have a lot of collaboration with a lot of companies. So for example, this is run in collaboration with a company to optimize this kind of bioprocessing. One thing, once you have your cell product, the most important thing before giving it to the patient, you need to do some analytics. So we have one lab which is only to do analytics. So let's say that I differentiate my cells into neuronal, but I need to make sure that the product before giving it to the patient has only neurons. So we need to do some kind of test to check that. So over here you have a cell sorter. So if you have different cell type, for example, you can sort, let's say you have neurons or you have cardiac, so you can separate each one in each tube. And of course, we have different kind of analytics. I'm pretty sure that you've heard of the -80 and the freezer things happening. You have a flow cytometer to check the identity of your cells. So not only you need to check the purity, the identity, and you need to check if it's safe for the patient. I think that's it from my side. Can take some questions.
That's absolutely fantastic. Thank you, Ron. I'm just jumping because I know Brenda's answering a question. So
yeah. So I think maybe just to say for the benefit of everyone and reiterate, you don't have to have a relevant work experience in this field. It's not, and depending on where you live, there may not even be a kind of biotechnology sector or a company that you can do work experience in. Think about the other ways that you can learn independently. Can you do an EPQ project? Can you do some independent research? Is there a science club? Are there events that you can get involved in? Citizen science, all kinds of things that will show your initiative, that will show that you have got a bit of get up and go, but it will also demonstrate your enthusiasm for science and curiosity about the world around you. I think that's more important than having spent a week watching someone prepare buffers at a pharmaceutical factory. It's great if you can get it, but also look at kind of ways that you can get more broadly involved in sort of science and technology. And there's many online events now with sort of learned societies, Society for General Microbiology and Royal Society of Biology. Check out their events, go along to them, and just show that you're kind of prepared to do things above and beyond and the normal stuff at school. I think that's what we're interested to see. But yeah, as I say, we're looking for people who are leaders, and there's many ways that you can do that outside of your A-level or IB program. We had a few questions asking about class sizes. So the target intake across the 3 programs, so BEng, MEng, BSc, and the 2 BSc routes, is 58 students. We typically have around 30 on the BEng, MEng route, and the remainder are split between the other pathways. It just varies a little bit year on year. But as you can see, that's a very small department that you're going to be joining in terms of, you know, sheer numbers. You will be in classes sometimes with maybe the chemical engineers. They're a much bigger cohort. There's over 100 of them. But you, as biochemical engineers, will be in your small class, and you will have kind of an identity that's very distinct. So sometimes you'll be in modules with other programs depending on what it is, but when you're being taught the kind of core modules of biochemical engineering, you'll just be with the other biochemical engineers. Okay. Any other questions? So why is UCL the only university? I think this is a historical reason. Other universities do offer programs. So you'll see quite often chemical engineering with biotechnology at a number of different institutions. We believe that to be able to do this interdisciplinary field well, you need to actually start both aspects from day one. So you need to go in with engineering, and you need to go in with the biology. A lot of other universities will teach the kind of engineering, and then much later on the biology gets introduced. We have a different approach. We're unique in that respect. There are kind of other universities that are interested in things like industrial biotechnology. They just do it in a slightly different way. And I guess this is a very much a uniquely UCL approach, because we're equipped with the facilities to be able to do this kind of teaching. And I think that's, you know, that's the reason why it's different. Oh, there's Mike behind me. Okay, there we go. So, if there are no other questions, what I would say is Kim has popped a link into the chat where I have done a kind of FAQ section of the things that I most commonly get asked as admissions tutor. So things about personal statements, things about preparing for the degree course, etc., etc. Have a look through those. If you've still got any questions, or if there's anything else that you want to talk about, drop me an email. And yeah, I think it's probably time for us to say cheerio. So yeah, Mike's here with me. Rana's probably still in level 5 somewhere, but yeah, thank you for joining. Thank you for staying with us, even though it's way beyond the time. I'm sorry for our late start, but it's really, really great that you're all here. And good luck with the rest of your studies. And yeah, I think just the message is that, you know, we're here and we would love for you to join us. But the main thing is, you know, you spend the next year really thinking about, you know, where you would like to go in future and what degree program really would meet your needs in terms of your career aspirations, but also what you want to get out of your time at university. So yeah, we're all here, and we're always happy to answer anything that you might have to ask. So good luck with it all. That's all I can say.
Yeah. Good luck.
Okay.
So thank you very much. I mean, huge thank you, Mike. Huge thank you, Rana, for that. And thank you, Brenda, for joining us. But most of all, thank you to the people who stuck with us. And as I said, there will be a link to this afterwards. What I'll try to do is I'll try to download the questions and the answers so that when we publish it, you can see those because not everyone had a chance to see it. So the links will be available. And you registered through UCL Radius. So come the spring, you'll get invited to our springtime lectures. We do a series of Spring into STEM lectures covering a range of subjects. We've done virus lasers, vaccines, cell and gene therapy. So a range of research and covering the modules that we do in our undergraduate programs. But keep an eye out for that, because we'd love to see you again. And as Brenda said, any questions, pop us an email. But in the meantime, thank you very much. Have a great day and hope to see you soon.
All right.
Thank you, everyone.
Bye.
See you soon. Bye.